Hello, in this video we are going to learn how to do sequence alignment with protein sequences. I have previously cloned my repository sequence alignment into the directory for lab 4. This repository is empty, although it has two scripts. We will use this script to count the gaps and this other script to generate our files. Before we start, we are going to go to the directory data and download 3 genomes. I will use the program NCBI genome download and download the GFF and FASTA file for each of the 3 genomes. Now we can see that we have created different files in RefSeq. We can use these commands to decompress the genomes. Now we can see that they have been decompressed. We can go back to our repository bin and edit our script. Notice how I have gene ID. This is only going to work for our human protein. For the other species, we will need to remove the gene ID from the script. If I want to download hemoglobin alpha 1 in humans, I will first need to declare the variable gene ID hemoglobin. We can also save our variables for FASTA file and GFF files, setting them to the human genome, and export these variables into the terminal. Once we're done, we can run bash and this will generate our files. Now, if we want to do the same for gorilla, we will remove gene ID and save our script and then search the gene ID for hemoglobin. We will also replace the variables for FASTA file and GFF file with the gorilla genome and export these variables using the function export. We will run the analysis again, but this time it will generate the results for hemoglobin in gorilla. We will do the same thing for the Drosophila gene GLOB1 and run the script. Now we should rename the header lines of the FASTA files to something more meaningful. For example, in this case, I will use the command set to substitute the lines for Homo sapien HbA1. Then we will concatenate the 3 files using the command cat into a new file called globincds.fa. Then we will use muscle to align the sequences. The problem with this is that for protein alignment, we will need to take into account the reading frame. To do so, we have to specify the codon structure. We can visualize the alignment using ALV. Another program to visualize alignment in the terminal is Alan. You can use the program tcopy to quantify the percent identity of the alignment. Now we can run the script count gaps. We can also use the command fattrans to translate the nucleotide sequences into amino acid sequences. We can also see the amino acid percent identity. We can also count the gaps and the stop codons in the sequence. Here, the X represents the gaps, while the C represents the stop codons. We can use another method that is translation aware, called TranslatorX. We can visualize our alignment also using e-links. You have to use square brackets to move to the right or the left in this program. Here we can see the percent identity. We can also see the number of gaps. We can do the same for the amino acid sequence. First measuring the percent identity and then counting the number of gaps in the alignment. This alignment has many gaps. This is because Drosophila GLOB1 and the globin's proteins in vertebrates split apart millions of years ago.